Today I'm going to be covering the synapse and neurotransmitters. So there are two types of synapses, electrical synapses and chemical synapses between neurons. Electrical synapses are faster, have faster reactions, are pH dependent, and have an ohmic connection. Chemical synapses are slower, but they're more versatile. Depending on what type of neurotransmitter is being released, you can have a different response. And they're also more modulatable than an electrical. So first, electrical synapses. They're made up of gap junctions. So gap junctions, as you can see, they're little places between the neurons where the plasma membrane is really close together and it's held together by these protein structures. They are made up of multiple units, and they're usually combined in little areas. And what happens is they're in two positions. They're either closed or open, depending on the protein configuration. And so these, when they're open, they allow ions to pass through these pores. I believe they're nonspecific. And so you get a response in the cell. So you have your presynaptic cell coming in, you have a gap junction, and you have what you're synapsing onto. So if you record from where the action potential is coming from and where you're sending your signal, you can see the first cell you have the standard action potential shape, looking over the appropriate time period, and you have a voltage change. But in the second cell, not all of that potential passes through. So you only get a little bit of a change. Here it's a depolarization of about 1. And this is called an electrical potential, EPSP. And remember, it's a lot smaller, but it follows the same general shape. The chemical synapse uses neurotransmitters to communicate with other cells, usually the one it's next to. So it has these neurotransmitters in the axon terminal in vesicles. And you can kind of see, okay, not very well, but there's little areas here where they're connecting to the plasma membrane, or where they're just floating around waiting for their turn. And in the diagram, you can see also some of them are little balls, little plasma balls sitting near the end of the terminal. And when these neurotransmitters are released from the vesicles, they activate receptors on the other side in an area that is called the postsynaptic density. Now, the good thing about chemical synapses is, depending on the type of neurotransmitter, you can have different effects. It can be maybe an inhibitory or an excitatory or something else entirely. But however, it's slow because you've got to wait for the neurotransmitter to diffuse. You've got to wait for it to be released. You've got to wait for it to diffuse across. It's got to activate something, and that activation can take various amounts of time depending on what it's doing. And there's two main types of neurotransmitter receptors. There's ligand-gated, where it's connecting directly with a channel, or there's secondary messengers, where it's starting a chain reaction that has some result. So how does the synaptic release happen? So here's a diagram pulled off the web. And you can see the very large vesicle, and the little pink are supposed to be the neurotransmitters, I believe. You can see a gap junction over here. So the action potential arrives at the synaptic button, and it opens the calcium channels. As the calcium flows in, there are proteins which cause the vesicles to bind to the outer plasma membrane and open. And as it opens, it releases the neurotransmitter through diffusion, and it goes across the very tiny synaptic cleft and binds to the receptors, where usually they open other channels, letting ions into the next cell, starting another action potential. And you see they're very complex interactions. It's not just one-to-one. It's not just in one area. You've got all the little terminals here. This is a electron microscope image of a synapse. I'm not sure which one is which direction things are going here. But they're all intertwined with each other. So depending on where the synapse is will have a different effect on the neuron and its response. So also remember that sometimes there are many axons going to a single cell, a single neuron. So occasionally the axon synapses with a dendrite and will try and generate an action potential down there to go to the rest of the soma. Or maybe it synapses directly on the soma. Or maybe this would be like for example, an inhibitory neuron. It's synapsing on another axon, maybe trying to stop that action potential from reaching the next cell.